Okay, so let's start. Today we are going to continue talking about modeling techniques. So this is the third session. In the first session, my idea was to introduce some tools that we have to model dynamical process, mathematical, computational, or both. And I show some of the assumptions that the different models have, and one could be more suitable to use one or another. And this is a summary. Today, fortunately for the people that is connected online, we have this pointer that I think that you can see it in the screen. So today it will be easier. So this is the summary of the different dynamical systems that we can use. And as we were discussing, it will depend on the system that we have, we want to analyze. And I also say that sometimes we can use different approaches to study the same system, because when we have a system that is spatially extended and we observe a kind of transition, it's very difficult to know which kind of transition it is. And although sometimes we can do some analysis or some computations to try to figure it out, and I will show one example later. But if we study a spatially extended system with the assumption of infinite diffusion with the ODEs, then we can actually know or have some evidence that, for example, this transition is a saddle node bifurcation, despite the system is spatially extended and the number of dimensions is very large. Okay. So the aim of the session today is to provide different examples of different types of models. You will see that in some of these examples that are mainly focused on biological systems, especially in ecological systems. But the approaches that I will explain will also be possible to use also in chemical or physical systems, because at the end, it is a matter of the interactions. At the end of this session, I will show two examples of my research in which we use models to explain data. That this is one of the goals of this course. When we try to explain data and understand the processes that are generating this data that we are measuring in the lab or we are taking in the field, we can use different approaches, as Luis was explaining the other day, the approximation with the splines or the fitting with different methods. But on top of this, we can try to explain some given data qualitatively or quantitatively. Okay, so I will give an example of each of these types. So I will explain an example in which we provided a qualitative explanation of the data. This means that we are doing some model or some simulations that is giving some of the results that we have observed experimentally or in the field, okay? But we are not reproducing exactly the dynamics. It's just that the model gives a similar phenomenon. And for the quantitative work, then this is about some experiments that we did in which we had a time series that was recorded in the lab, and then we did some model, and then we fitted the model to the data. So the last system I will show you are those that are specially studied in this course. So we have some data, we have a model, and we have to fit the model to the data. Of course, here we are also explaining qualitatively the data, but we are quantifying some parameters. Okay? The other approach, the qualitative, it's like, well, you will show clearly. Okay, so one second, that this is. Okay, so I will start with a very used system that it's a metapopulation. Okay, metapopulation is a is a term that was studied or started studying in the 70s, like last century. And a metapopulation is a group of populations which are usually called subpopulations that occupy spatially distinct patches. So they are physically separated, but they are close enough to support processes of colonization and extinction. Okay, so you have to imagine a species that is distributed in different patches that are separated, but they can invade or colonize the different patches, but they are spatially separated. Of course, we have natural metapopulation, natural fragmentation. For example, we have a forest that has suffered some fire, or we have a river crossing a place, and we have different patches that are separated. And of course, we have the anthropogenic fragmentation. So of course, these systems are very, very difficult to model because we have, this is a little bit crazy. Sorry. We have, okay, so we have a quite large complexity here in these places because we have a lot of different patches that are heterogeneous, and we can try to model it. Of course, we can go from the most simplest cases where the patches are symmetric, and then we can extend the complexity. So the metapopulation concept was coined by Richard Levins. This is the famous Levins model. And he used it to model populations of insects in the field, okay, and the model of insect pests in these fields. Okay, this is a very old paper. You will see that the model is very simple. Actually, the model will be very familiar to you. So yeah, mainly the dynamics of metapopulation is governed. I mean, the dynamics of the whole population is governed by colonization and extinction processes. Then we have that each subpopulation, it's like in some way independent, but they are connected through dispersal. And in this type of systems, we have that we can have, for example, local extinctions, but the population as a whole can survive. Okay, so we can have local extinctions, but since we have this colonization process, the population will persist. Also, I have to say that most of the epidemiological models, the SIR, the SIS, and different versions are also metapopulation models because they are compartmental models, and we have, like, let's say, the colonization processes is like the transition from one epidemic state to another. Okay, so metapopulation theory was first developed for terrestrial ecosystems, but since some decades ago, it has been also applied to marine ecosystems because, of course, in the ocean, everything is mixed, but we have some individuals that can have colonies, for example, the sea urchins or the corals, okay? And they also can occupy different places on the space, and then water can move the larvae or the eggs, whatever. So this is a diagram in which we have two axes. This is the degree of connectivity, and this is the variance in patch size, because there are a lot of different metapopulation models. We have these highly isolated, mostly small patches. It's called non-equilibrium, in which we have independent subpopulations, which each one with its dynamics. And then as we increase connectivity, we have the typical metapopulation in which some of the patches are connected with some others, but there are some others that are not connected, so we do not have full connection. For example, here the green patches are the occupied patches, and the white ones are the empty, and these are the dispersal directions. And then we have the panmictic. Panmictic means that all patches are connected between them. And then we also have some other examples, like the dissected landscape, in which we have, like, let's say, a single patch, but that has some barriers inside, and species can colonize. And then we have a very famous model, which is the Iceland mainland model, in which we have a main source, and then we have some islands that are receiving this population. This kind of systems, usually the population in the bigger area is persistent and is abundant, and it's very difficult to become extinct, but then it has these reservoirs. Okay, so when we have to model a metapopulation, we have different approaches. For example, because here, this is a kind of system in which we have a space, in some sense, space, we are modeling a spatially extended system, but it's different of a PD, of course. So the Levin's model is the simplest metapopulation model. You can see how the model looks like. This is very familiar to all the people. So this is the logistic equation in a time continuous approach. C is the colonization rate, and E is the extinction rate. So here we are modeling a metapopulation with a single variable. And this is why here I put explicit space in an implicit way. Why? Because this model considers the fraction of occupied sites. So how many sites in a given metapopulation, actually with infinite sites, are occupied or not. And this is a very abstract model. So this model says that n is the fraction of patches occupied at a given time. During a delta t, each occupied patch can become unoccupied due to an extinction, of course, with probability E times ddt. And this is why we are using here the logistic equation, 1 minus n are the patches that are not occupied, or the normalized fraction of patches that are not occupied. So if we assume a constant rate here of propagule generation, so C is the colonization rate to other patches, then, of course, this equation, this n, will grow with the growth rate, or let's say colonization probability, Cn ddt. And the n dt, of course, is the rate of change of the number of occupied patches. So this is a very abstract model. You know that this kind of system has, let's say, simple dynamics. So we have two equilibrium points. One is everything empty, and another one is that there are some empty, some patches that are occupied. And after we overcome a threshold that takes into account some relation between C and E, this system suffers a transcritical bifurcation, and then when extinction is very large, the only stable point is full extinction. So what are the assumptions of this model? And this is very important because we can use the logistic model to model different things. For example, how a population of bacteria grow in time. And the assumptions of this model, in which we have the growth rate of the bacteria, the growth rate of bacteria and the decay rate of bacteria, it will be different depending on the system, although the mathematical model will be the same. So the Levin's model assumes that there are infinite patches and that the deterministic approach works, because in deterministic systems, we do not consider noise, and then we assume that the size of the system is infinite. We also assume if they are occupied or empty. So it's a fraction of empty or occupied. Here we do not have, let's say, a population of individuals. Is if the patches are occupied or not. It assumes that local dynamics do not affect colonization and extinction rates. Also, that local dynamics occur on a much faster time scale than at the metapopulation. And then this is the colonization extinction dynamics. And immigration has no effect on local dynamics. It also assumes that there is no patch structure. I mean, these patches have not a geographical position, a specific position. It also assumes that patches are symmetric. So if we wanted to do a system in which patches are asymmetric, then we should modify this model. We could not use this. And this is what I was explaining in the first session. So here the processes occur by mass action. Okay, so remember that mass action law says that the rate of production of a product, let's say, or the rate of synthesis of something, the rate is proportional to the reactants that are conducting this reaction. Okay, so the more reactant, the faster will be the reaction. Okay, so this is very simple model. Has been studied a lot, you can imagine, a lot. Now I will show you, let's say, this model in which we have added one parameter, that is this D here, that this is the Levin's model with habitat destruction. So D is the fraction of destroyed patches or destroyed sites. And it means that if D is equal to 1, it means that all the patches are destroyed and population will become to extinction. Of course, this model is very similar to the other one, and here we can observe the same of the other one. Here we have this critical value of a fraction of the habitat of this is also called habitat loss. This D is the fraction of habitat destroyed or habitat loss. So this is the relation that separates the scenario of survival from extinction. So if D is below this value, we have survival. But if D is above this value, we have extinction. And this is especially this curve here, that it's a transcritical bifurcation curve. Okay. One thing that we can do when we have one-dimensional systems with ODEs, we can compute the potential, and it's very easy to compute. So the potential is minus the integral of the vector field ddx. And this, you can compute it most of the times analytically, and what you obtain is this value of the potential as a function of x, and then depending on the parameters, where here you have this capital gamma, where it depends on all the parameters, it gives you the potential. For example, when D is very small, we have this potential in which we have the zero, the extinction of the population, which is at the top of a peak. So this is an unstable point. And then we have this point here that is the equilibrium point of the system that is stable. And as we increase E, this potential changes. And finally, when D is 0.995, we only have a single valley at the origin. Okay. And this type of analysis help a lot in understanding the dynamics. So now I will explain a work that we did some time ago, and I am explaining it because here we mix different modeling approaches in order to understand the system more in depth. So this is the Levin's model, the same model with habitat destruction, but if you can see here, we have the x square. And this is a model in which we are considering catalysis or facilitation. Okay, if we write the stoichiometry of this growth term, we have one individual that meets with another one at a rate c, and this gives 3a. This is an autocatalytic reaction, as a difference from the exponential, in which we have a that gives 2a. Okay, plus some whatever. So if we want to do the mass action with the law of mass action, the growth term of this, if x is the amount of a, then this will be the product of x squared. Okay, so this is the growth function. With this not exponential. This growth function is hyperbolic. And this growth function, if we compute the solution, This is exponential, but this one is not exponential. And with this type of growth, we can go to infinite time, not for this case. Okay. But anyway, here we also included the logistic function, which is mainly that the population, due to competition, is affecting the growth rate. This is why we're multiplying by minus x. So this is the effect of competition between the individuals, let's say. Here we are not applying the assumption of the patches. So this is like the population, okay? And this is the fraction of habitat destroyed. So what happens, and this is something that has not been investigated a lot, what happens if we have habitat loss and we have some species that cooperates, that the individuals cooperate? Or, for example, it is known that under some conditions of water availability, plants undergo processes of facilitation. This means that these are plant-plant interactions, and it means that if you are in a place where water is available, there you will have large amount of plants, and these plants will get benefit from the plants surrounding because we'll be able to retain more water, or they will be shadowing and will be avoiding evaporation. Okay, so this very, very simple model, it's a model where we have facilitation, habitat loss, and competition. Of course, this term here, x squared, changes the dynamics completely. Here you have the reference of the article. It's published in Population Ecology 4 years ago. Here we have 3 equilibrium points because this system here, in some sense, it reminds a little bit with the normal form of the saddle node bifurcation. And here we have this, we have the fixed point that is the origin, as you can see, x0, but also we have these 2 equilibrium points in which we have the fixed points with the equilibrium points with a discriminant, so we can suspect that we'll have a saddle node bifurcation. And this is what happens. So here, the transition to survival to extinction is not continuous, like the transcritical bifurcation is catastrophic. And this is what nowadays ecologists say this is a tipping point. Okay? A tipping point is when you have bifurcation and you have this point, you have this turning point, and you have a tipping point. And this is what they refer to discontinuous transitions. As a difference from the Levin's model with habitat loss, here we have bistability. It means that the origin is locally asymptotically stable, and this is very easy to see if we do the derivative of the field evaluated at the origin. The value we obtain is minus epsilon. It means that it is an attractor. Okay, and this means that when we are below the critical D, if population is not large enough, the system will continue go to extinction. And this is very important for ecological systems. This is the potential, also computed with this formula here. And here we can see that when D is very small, we have 2 very well-defined wells that are actually describing this situation in which we have one stable equilibrium involving survival of the population and another one involving extinction. If we increase D, this well here starts increasing, and at some point, it turns out that we only have one single well, which is extinction, and here we have the saddle node bifurcation. Okay, this is very nice, but in ecosystems, the species are spatially extended. Okay, so the mean field model is telling to me that the system will have saddle node bifurcation, catastrophic transition, whatever. But we are interested in the impact of, or in the dynamics of these systems when we have, for example, plants or shrubs. They are spatially extended. And I want to analyze the spatiotemporal dynamics of this system. One of the things that I can do is to implement a cellular automata. So for the cellular automata, what we consider is that we have a grid, a square grid, and we have periodic boundary conditions. Okay, this is of course not very realistic. Usually when we have an ecosystem, we assume that the border is closed, but here we consider periodic boundary conditions. And now I have to implement this reaction programming in a computer program, in a cellular automata. And this is a nonlinear interaction. So how can I introduce it in a cellular automata? So the only need that you have to consider is that this is density dependent. Okay, this process is density independent. I have A and it reproduces to A. But here we need another individual of A. And this is density dependent. This is why I have this product here and the nonlinearity. So how would you do that? You can do it many ways, but usually what we do is that when we implement the state transition rules, if you remember, we choose a cell at random. We choose a cell. Imagine that this the central one. Then we choose another random neighbor. Here we have the more neighborhood, 8 neighbors. And we have the rules that depend on the neighbors and on the site that we are choosing. Okay? So here what we did is the following. These are the state transition rules. With these 2 state transition rules, I am simulating a system with these dynamics. With these 2 state transition rules, I am simulating this thing, and this thing. And this is already implemented also in the automata. So we define a function, state transition rule, reproduction and colonization. So, sorry. We select a random site. If this site is empty, nothing happens. If the site is occupied, then we select another random cell in the more neighborhood. Okay, we select a random neighbor. So if this is this as J, okay. So if the neighbor contains another species, the species that I have chosen will reproduce and colonize another random neighbor. And this is how I am modeling this interaction. So I choose a site, I choose a random neighbor, and if it is empty, then this guy will make another individual. And this is exactly this kinetics here. Then what we consider is that if the new individual that is produced, that is put in another random site, goes to a place where there is an individual, nothing will happen. The idea is that this individual colonizes another place where there is another plant and has no resources for growing, and it dies. Or if it goes to a destroyed site, it cannot grow because the soil is destroyed. There is nothing. Okay, it cannot grow. And then extinction is very easy. Extinction, it's an exponential term, is non-linear. So I choose, I don't need to see the neighbors. I choose a site, and if it is occupied, I remove this individual with probability E. Okay, but now here I'm missing something. The logistic term. How am I introducing the logistic term in the automata? Very easy. I have a finite lattice that will have a carrying capacity, because when all this lattice is full, or said in other words, when the population starts reproducing and starts filling the lattice with individuals, then if we make a plot of this occupancy dynamics, we will have a slowdown with the population. Okay, and this is exactly the logistic. So cellular automata program like this have a kind of logistic like behavior. And I will give you some evidence of this, because you could say you are saying it, but we don't know. But I will give some evidence. Okay. So This is a very interesting. Now I'm going to explain a very interesting thing that I found in a paper that was about viruses, about virology, and it's that we can play with the neighborhood. So usually, most of the cellular automata models take into account the closer neighbors, okay, the closer neighbors, or the second order, let's say, neighborhood. For this work, because this work was a work that was developed to investigate the effect of habitat loss in ecosystems where you have facilitation, and these are mainly plants. But plants can disperse the seeds very far away. It is not necessary that they disperse the seeds just in the surrounding. So what we did is to use this paper here, in which we have a dispersal function, which is this one. Okay, so this function provides a random number between 1 and m that will be, let's say, how far is the neighbor. Okay, so the number 1 will be that I am choosing my first neighbor. Number m is that I choose the neighbor that is m times far away from me. This is some function that you can implement in your automata in the computer, and then you can make the colonization rate to depend on this function, and then you can say if the seed will go just next to me or will go very far away. But this go very far away depends on the distance. So the more longer it has to go, the more difficult. But of course, it will depend on the parameters of this function. So, for example, delta is the number of this function that will say how far you are going. M is the maximum distance that you can achieve. Then R is a uniformly random distributed number. And this is a very important parameter. K determines how steep this distribution of delta is. Okay, now I will show you some examples. So we have like two extreme cases. If K is equal to 0, okay, if K is equal to 0, then if you have a look to this function, then the distance is 1. Okay, this is very easy to see, and it means that this probability of going, or the distance that you can go, declines very fast. So the probability to go to a distance declines so fast that you will only move to the nearest neighbor. If K tends to infinite, or if you make K to increase a lot, then this effect of a decrease, the probability of moving decreases in distance, disappears. And then you can move to any distance with the same probability. And you have a uniform D. Okay? So it's better an image than 1,000 words. So this is this probability of going to a distance delta as a function of delta. So if K is equal to 0, then you will always go to the first neighbor. If I increase K, then you will move, or you will colonize farther, but depending on the distance. So it will be more probable to go to be very close, and it will be more difficult to go very fast. And this happens when you increase K. Okay, so for example, for K equal to 10,000, this is just a numerical approximation. Here we have choose M to be 100, okay? You will go anywhere. So you will have the same chance to go to the first neighbor or then go to the further one. So what K would you imagine for this model? What is this model? What K is this model assuming? Which of these panels is this model assuming? Here we don't have a space. Everything is well mixed. So in practical terms, it means that I can go anywhere. Because if I am using this one, this means that the dynamics will depend on the closer neighbors. But here, no, this is a mean field model. This is completely mixed. So if I use this distribution here, that the probability of colonizing is everywhere, then I am closer to this system. Okay, because this would assume that the lattice is infinite, etc. Why I'm saying it? Okay, because one thing that you can do for many reasons, the first one is if the model is correct. So I did a computer program. If random blah, else random qual. And if I don't know, whatever, then if do while, I don't know. Okay? So am I simulating this system as correctly? It's very easy. For this system, that is easy. For others, it's very difficult. So what can I do? I just turn or convert this automata, okay, to a mean field system. Then what I only need to do is to break the correlations. So instead of saying, if I take a site and then I take a random neighbor, and if there is here an individual, then I can colonize a close individual, or I can move more or less in this place. I break correlations. So what I do is I choose a site. Then I choose any other random site of the lattice, any. And when this new individual is produced, I put it in any other random site. So everything is mixed. But I am keeping the interactions, the density-dependent interactions. That is this reaction. So if I do this, of course, I should obtain something like this. No? Because I am facilitating these nonlinear reactions, completely mixed. Okay, this is the ODE. Let's have a look. This is the ODE. And you can see that the turning point, it's about 0.3, more or less. Okay, let's do the simulations. Let's run the simulations with the replicas and all the things. And, oh, surprise, this is what we get. This is a population increasing the fraction of habitat destruction, like in the mean field model, and at about 0.3, the population collapses discontinuously. So this is the first thing I did, just to see that if I was implementing the system correctly with this thing. Okay, so here just a detail is that if you run a simulation very close after the saddle node bifurcation, but of course the saddle node bifurcation is taking place here. This is another thing. Okay, we can say it's a first order phase transition, but it's a catastrophic transition. We can see that when we are very close to the transition, we have this dynamics that is very, 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 very long, and then we have the collapse. And this is a ghost. If you remember in my first session, I explained that. So here you can see the ghost, and the ghost arises from these things. It's not a mathematical effect. It's an effect of the kinetics that I can identify with mathematics. But the ghost is there, as it was in the experiment with the electronic circuit. This is a real thing. It's a real ghost. It's the first evidence of a real ghost in the world. So, okay, but usually space can have a very important effect on dynamics. And there are a lot of examples in the literature. When you consider space, things change. How things change here? For our surprise, when you consider space, so now I have not broken the correlations, so everything is happening in some neighborhood. When I consider that colonization goes to the first neighbors, these are for different fractions of habitat loss. For example, the first arrow is for a value of habitat loss about 0.25. And you can see for different times that you have spatial patterning. You have patterns. Patterns that change, but they remain more or less the same. And this is the time series of the population. No, good question. So here what I do, usually one approach that you consider in the literature is that your time, so this is your time, okay, for, let's say, number of iterations, whatever, okay, because this is a discrete system. And here you have the state transition rules. Here you have the colonization, reproduction, and extinction. Okay? And this is time, this tau, okay? Of course, one question is how we put these two state transition rules at work. Because now there are two, but I have programs on automatas with 8 state transition rules. And you have to be very, very careful, because if you make these things to happen in order, you are introducing biases. So usually what I do, you can do two things. And one of the things I just learned 2 months ago. So one thing is, usually what I do is I choose them at random. So then you choose one at random, and then if imagine that you choose it, then with some probability, here C, okay, this rule will occur with some probability. If you choose this one, so they have the same probability, okay? And then if you choose this one, so this will happen with some probability. What I do, it's called asynchronous updating. Because if my time booklet or my time loop, sorry, I use this way, it will go very, very, very slowly, and you need to iterate a lot to go to equilibrium. So what I do is that here, put another 4, and here, if L is the size of the lattice, I do L squared times this process, and every time I do this process, I choose a random cell. So of course, in a given time iteration, I can choose, if I do it L squared times, I can choose the same cell twice. As larger is the lattice, the low is the probability that it happens. But on average, if you iterate during many generations, things compensate. And this is a very way to do so. And this is exactly how I did. So here you have this tau. So at each value of tau here, that you have the point of the population, you do L squared times the actualization of the lattice. You can do one by one, but if you do it randomly, you could also do it sequentially, but then you need to take into account some other things. Because of the neighbors, you need 2 lattices, then you have to update one lattice, and then depending on it, so you have to keep the neighbors. So here it doesn't matter. Everything is random. And so if I increase the fraction of habitat destruction here, 0.3, the population diminishes. Here it gets like stationary at 0.38, here 0.35. You can see that the patterns change and there are more holes. Here I have to say that the empty sites, so I should set it at the beginning. This is a cellular automata with 3 states: occupied, empty, and destroyed. Destroyed is in red. Empty is in white, and occupied is in black. And to introduce this D, what I do is that at the beginning of the simulation, I randomly place the fraction of reds that this parameter is saying to me. So if this is 0.5, before starting the simulation, I randomly place the half of the sites destroyed. And then I put the individuals, keeping these red sites, because these red sites have to be there, fixed, and then you iterate. Okay. It's very interesting because when you go to the field and you go to the nature, you see that vegetation makes these patterns. And this is a very simple model where we have facilitation. There is a paper, there are some papers published, these are from the 10 years ago, 15 years ago, published in, Nature Science, whatever, that they were able to reproduce spatial patterns in the Mediterranean, in the Kalahari Desert, in Greece. They were able to reproduce the observed spatial patterns with very simple cellular automata models, because the main drivers of this system are water and facilitation. Okay, and when we are very close to the transition, the population, of course, gets very, very small, and you can see like these isolated patches of vegetation. This is for automata of size L200, so 40,000 cells. And you can see that the time series are quite noisy. Okay, so does anybody see something here strange compared to the previous slide? Look at the bifurcation diagram. The transition has changed. Now it's not discontinuous. It's a smooth transition. And the system is autocatalytic. And the saddle node bifurcation has disappeared. Or I don't know what is happening. And actually, we don't know what is happening here yet. What we know, and this is, I think, some of the more interesting thing of this paper, is that which neighborhood you consider matters, is important. Why? Because of facilitation. If I can put my little daughters, my little plants surrounding myself, facilitation will take place, because I will be able to retain water and my little plants will be happy. But if this k, and this is why we use this function k, if this k increases, okay, here you have an example with k0, that this is actually a zoom of this smooth transition. If k is 6, I mean, you can go to the first neighbor, second neighbor, third neighbor, fourth neighbor, fifth neighbor, or sixth neighbor. And it's more probable that you go to the first, but it decays with the distance, then the transition changes and it's discontinuous. There is one article in PNAS that they saw something similar, but the reason why this is happening, it's not understood. I mean, the bifurcation is different. This has changed. So one thing that I would like to do, but I don't know how, is to make a model like this, mean field model, but in some way introducing the effect of locality. Perhaps it's not so difficult to do, and then it would be possible to see what happens to this bifurcation. One thing that could happen, that I have thought about it, is that here you have the stable branch. Sorry, 3 buttons. And here you have the stable branch, you have the jump, and here, of course, you have the unstable branch. And it is there because I computed it with a cellular automata in a very simple way, and now you will see there is a stable and an unstable branch. One thing, the simplest thing that, in my opinion, could happen is that here, this is the stable branch, and the stable branch is here. It's out of the, let's say, positive numbers, or octan or whatever, okay? But this is just speculation. This is not understood why this is changing the bifurcation. And this is very important in ecology, of course. If you have this transition, you can say, you go to the field and you say, wow, 40% of the surface is full of plants, everything is okay. Yes, but if we have a very tiny increase, the population collapses. And this is something that happens in nature. There are a lot of examples. And actually, in a few weeks, we will have a review out explaining these things and giving a lot of examples in nature, in coral reefs, in forests, in savannas, in marine systems, in ecological communities. There are a lot of examples in which these shifts have been observed in real systems. Okay, well, these are some other analyses, and this is something that it's interesting because this is a cellular automata. Since it is a simple one, we can do something. So here what I did, this is the same like before. So I am increasing K. And here you go from more or less an abrupt transition, but as you increase K more and more, the system resembles more to the mean field model. Look at this one. It's exactly the same diagram. So how I computed the unstable branch with the automata? Very easy. You fix the value of D. For example, here, this one. Imagine you fix the value of D to 0, 1, then you make a program, and you go, you investigate different initial conditions. You increase the different initial conditions. And the first group of initial conditions that go up and do not go to 0, then you record this point. Here you have the point with some error, okay? And by doing so, this is very easy to do with a cellular automata. You just need to play with the initial conditions. So let's say all the initial, if you see these two arrows, all the initial conditions above 0.1 more or less will go up, because this is the natural dynamics of the system. There is an attractor here. This is an attractor. But this is a simulation I did. And I am simulating some interactions. And these interactions, I can represent these interactions with a simple equation. And with this model, we had a lot of information. The only thing is that we do not know what bifurcation we have when the neighbors are first one. Okay, I don't know if you want to make a 5 minutes break or we can go on. Okay, so we'll go on. Okay, now I will, so now we have been so far talking about metapopulations, and now I will talk about cellular automata. In my previous example, I have used a metapopulation model, okay, because in some sense I can consider that it's not the same, because these are not the fraction of occupied sites. Well, I could say that these are the fraction of occupied sites with two species, but it's not direct. But this is a metapopulation model, and I extended this model with a spatially explicit here. Space is implicit here, okay? Because we have this logistic term that is modulated by the habitat destruction. The larger the habitat destruction, the lower the carrying capacity. It's like if I'm doing the lattice small. But I used a cellular automata. So cellular automata is a discrete dynamical system, is discrete in space, discrete in time, and discrete in variables. It's a fully discrete system. And these are also usually called individual-based model or agent-based models, because they have discrete variables. So usually a cellular automata, there is a lot of mathematical theory of cellular automata. I mean, mathematics with theorems, whatever, about cellular automata. And the computational, let's say, development of cellular automata was done in the 80s, late 70s, 80s, because of course, of the computers, by Wolfram, Packard, etc. Okay? They established the basis of cellular automata. Cellular automata are very powerful, as I will show you now. So mainly cellular automata consists on a finite set of symbols, which is the alphabet. We can have a binary alphabet, 2 symbols. And then we have a local function, okay, such that the output of this function f small depends on some cells. Usually what we have is that we have that this function depends on a given site and then on the neighbors. And this is where we have the rules, the state transition rules, that will be the rules that will say that depending on what you have in the site and on the neighbors, the automata will evolve as I want, differently. Okay, so a cellular automata consists of a state space, which this is a discrete and is a space. It's if we want to simulate a physical system, for example, a crystal growing in a dish, a salt crystal, or we want to study how a population of bacteria spread on a petri dish, then we will use 2D state space. If you want to model the growth of a solid tumor, we will use 3D space. Then, of course, we can use more dimensions if we are doing more abstract things. So usually we have one or more state variables. In the case I showed you about the plants, we have empty, full, and destroyed. So we have like 3 states. State variables 1, for example, that is the number of plants. Then the state transition rules, that is how the automata evolves. And of course, we can decide some degree of locality. We can use the first neighbors. We can use 4 neighbors, the von Neumann neighborhood. We can use 8 neighbors. We can use hexagonal neighbors, whatever. And boundary conditions. And the initial condition, the initial configuration. Okay, the cellular automata are also named cellular spaces, tessellation automata, iterative arrays, cellular structures, whatever. And usually all what I said before, that it's discrete, I will also talk about another system that it's called couplet map lattices. These ones have been studied a lot to make research about spatiotemporal chaos. And they have also been used in problems in ecology, but you will not find a lot of literature on these approaches. And I will explain later what they are. The difference is that couplet map lattices are the same that cellular automata, but the variables are continuous, are not discrete. So the first automatas that were done and were studied are called the elementary cellular automatas. And here we only have 2 states, 0 or 1. These are binary automatas. So the dynamics at t+1 depends on the state of the cell T and on the 2 nearest neighbors. So here we are looking left-right neighbors. And there are a lot of different automatas. This is one that you can also represent the automata by this way. So you have the current pattern. This is the central state and the neighbors. So here we define a function that changes the bit in between. For example, if the sum of the neighbors is odd or pair. Very simple rule, but this gives you a rule of evolution. And with these simple rules, and there is a very nice book, this automata gives this pattern. And this is a marine snail that lives in the Pacific that is very poisonous. And it's very beautiful. So how can we get this pattern? Very easy. So this is a 1D automata. Here we have the state space. Okay, we can have, if we want, a toroidal boundary condition. So the one moving this way will appear here. This is space. And this is time. So this is iteration 0. So at iteration 1, what I need to do is to take the value of each of the cells, look at the neighbors. In this case, I would look this state, this neighbor, and this neighbor. And according to what we have, here we put 0 or 1. And you iterate it on time. This is an example of the automata that is called the rule 30. Rule 30, this is a nomenclature that refers to the decimal number of this binary sequence. Okay, the output of the function. This is, if you compute here the decimal, the integer, it's 30. And this rule 30, when you iterate 20 times, you obtain this pattern. But if you make a lot of iterations, you obtain this kind of pattern. And here you can have chaos. Chaos in the sense of, well, I will not say the Van-A, this would to say too much, but that you have sensitivity to initial conditions. So you change a little bit the seed, the automata is very different. So for the one-dimensional cellular automata, or the elementary cellular automata, this is one-dimensional with binary states, there have been defined 4 classes. Class 1, the evolution always go to a homogeneous final state. It's like an equilibrium value or fixed point. Class 2 usually gives periodic patterns which are separated, but they, at some point, the automata structures itself and it gives you bands that remain forever. It's a kind of periodic pattern. Class 3, like this one, rule 30, exhibit chaotic behavior and aperiodic patterns. Actually, this one, this automata class 3, I think MATLAB has a function that generates pseudo-random numbers using this automata. Okay. And class 4, it's like a mix of the previous ones. Why? Because you can have, let's say, ordered and irregular structures, but you have like formation of macro scale patterns that interact each other. And it's better an image than 1,000 words. These are the 4 classes. So this one, you start with a random configuration of 0 and 1s, and you achieve homogeneous state, class 1. This one, you start with a random configuration, initial configuration, and you got periodic patterns. This one, it's class 3. You start with random, and you got here, this is like Sierpinski triangles mixed. And it's interesting. So last year I was supervising a master science thesis about one student from master here at UAB. And he did a work classifying this automata, but with other criteria. I mean, with Lyapunov exponents, with entropy measures. And it's very nice because these class 3 systems, you have many. You have the rule 30, the rule 96, the rule 241, and they belong to the same class. Okay? These are like universality classes. And this is class 4, that you can see that you have, for example, here the formation of this structure so this is a kind of macro scale pattern that interacts with this one, with this black or the white, and then it changes and gives place to this other structure. So you have like interaction of patterns. Okay, if you have a look to the literature and look for modeling of problems with a cellular automata, you will find a lot of things in physics, chemistry, biology, a lot of things. And some of these automata give the very similar results to experiments of aggregation, nucleation, spatial patterning, etc. So now I will very quickly explain this. This is one of the first paper about 2-dimensional cellular automata. So now these are not elementary. These are 2-dimensional patterns by Packard and Wolfram. And here, well, these are some general things about these functions that I was saying. These are the neighborhoods, for example, the von Neumann neighborhood in 2D, of course. Consider the 4 nearest neighbors, the 8 ones. And from my experience, when you do one neighborhood or the other, it's quite similar. I mean, you don't see significant changes. Okay, then we have different examples of 2D automata. We have the totalistic cellular automata, in which the value of a given site at time t plus 1 is a function that sums all the states of the neighbor. Okay. Another one is the outer totalistic, in which the value of the site at t plus 1 is a function that depends on the site itself and the sum of all the neighbors. So here we have a lot of different options. And actually, in the simulations I was showing about the plants, I chose those rules because these are the rules that are expected to occur in the ecosystem, that you have facilitation. Then you have to consider this density-dependent thing. Okay, boundary conditions. So a cellular automata, by definition, or let's say in terms of mathematics, assumes that you have an infinite grid of cells. Of course, when you go to this, is like the PDs, when you go to the computer to make a cellular automata, you have to consider a finite grid, and you need to define the boundary conditions. So there are a lot of different conditions. For example, the periodic boundary conditions, in which for 1D you have a ring, and for 2D you have a torus. You also have reflecting boundaries that you consider that the cells outside the lattice reflect, are like mirrors. Because one thing you can do is that you can assume, because cellular automata are very plastic. I mean, I can do whatever I want. In some of the works, the one that I was explaining in the first session with the hypercycle, I defined the automata considering that in a cell we can have two different species, and they interact when they are here. But this is actually the same like if I have one here, and I choose these two cells. Okay, so more or less it's the same. I can put here 3, 4, 5, whatever I want. Okay, usually you only put one per cell, but this is up to you. Another one is to consider that there is no boundary. Then you have to work with dynamic memory programming. Is that if you go here, then you add another, so the automata grows. Okay, this can be also, for example, if you have a growing tumor, you can have this growing, that the state space that is formed by cells is growing as time goes on. Yes, we have also hexagonal cellular automata. We don't need this automata to be a square grid. It can be hexagonal. We can use, this is something I've never done. Well, I did, but just to make some experiments, is that when you have your lattice, imagine a 2D lattice, you have your state transition rules. You have a set of state transition rules, for example, set 1 and set 2, and here you have a given dynamics 1, and here you have dynamics 2. And dynamics are different. For example, here competition is strong, and here competition is small. And I can say, okay, I will apply state transition rules 1 here, and then I will apply state transition 2 here. And this is a way to introduce heterogeneity. And this is very useful to study, for example, processes in landscapes, heterogeneous landscapes or heterogeneous tumors, whatever. And of course, we can use, we can also program deterministic or probabilistic rules, deterministic automata or probabilistic cellular automata. Okay, this is one of the most, let's say, nice examples. This is, I don't know if you know about the Belousov-Sabotinsky reaction. This is a biochemical reaction that it's not a simple reaction. I mean, you have all these reactions. Okay, you have acid malonic, bromate, etc. Water. You have a lot of things. Okay, and these are the number of reactions. This system is very easy, and actually you can do it in the lab. You only need a Petri dish, mix all these chemical species, and here you have this paper here, and this is how the reaction looks like. This is a real experiment in which you have traveling waves, okay, that interact with each other. So it's very nice because some of my colleagues did the experiment in the lab, and when you mix all the species, you get like a red homogeneous pattern. But then if you click with a pencil or with a clip or whatever, you have this perturbation, and then the reaction starts oscillating. And this is an example of a self-oscillating chemical reaction and has been studied a lot. And actually, there are works about the spatiotemporal chaos. They have computed Lyapunov exponents. They have computed Fourier transforms. They have done a lot of research. So what happens if we model these reactions, all this set of reactions, in a spatial extended system? Okay, we can do it with PDEs. We use the stoichiometry here, like the same like this. We use the stoichiometry of all the reactions. We build the differential equations, and we use PDEs. And this is what we get from the PDEs. And you can see that with the PDEs, you also have this patterning. Okay. If you do the same with a cellular automata, you can also see. This is actually the paper I was showing you. This is Belozov-Zabotinsky with cellular automata models, and this is what you get. These are like spirals, okay, that move on time. So this is the experiment with the chemicals in the lab. This is PDEs, and this is cellular automata. Okay. Any question? Any comment? Any idea? Because now I'm going to switch to the couplet map lattices. Very short explanation. So all these things are like tools that I am showing you that they have applications for modeling. So couplet map lattices are like PDEs and cellular automata, dynamical systems with explicit space. As cellular automata models, they have discrete time, discrete space. I was showing grids, I was showing lattices, but you can use networks also. Okay, and the state variables are continuous. This is the only difference, and they have been very used to study spatiotemporal chaos. So this is it is said that they were invented by Kaneko in the 80s, and they have been used to study complex phenomena in solid state physics, optics, chemical reactions, and well, possibly in biology, because these guys were at these times were wondering if these systems would be useful to describe biological processes. Here you have some articles that have used couplet map lattices. There are not a lot. There are few. There are few papers. It was mainly at the end of last century that people was very interested in these systems. And the idea is very simple. So here what we have is that we have a function or a system of equations with continuous variables, and we coupled them in a discrete way in space. So everybody recognizes this plot. This is the logistic map with all the dynamics that we have here. Transcritical bifurcation at R equal 1, the flip bifurcations, period doubling, then we have the chaotic region. Period 3 implies chaos, chaotic intermittency, more or less here, and all this stuff. Okay, chaos. What happens if we use a couplet map lattice? So here we have a couplet map lattice in which we have a discrete time system, n+1, x is continuous, and we have a coupling function. So this is the logistic map at site S, with a parameter epsilon in front of. And this is the left neighbor, S-1, with parameter 1 minus epsilon. It means that if epsilon is 1, we have a uncouplet system. We have two logistic maps in site S and site 1, but they are not coupled. If epsilon is smaller than 1, then we couple the system. Does it change the dynamics? Yes, this is from Wikipedia. Yes, it changes the dynamics. This is a completely uncoupled here you have to imagine like a number of rows with different cells, and we have this function here with epsilon equal to 1. Then these are like different logistic maps. And here we are coupling these maps with using, I think it's epsilon 1/2. And you can see that from complete chaos, we have the emergence of order patterns. Okay. So one of the most known couplet map lattices is this one. This is a general function in which we have a coupling of, let's say, a given cell at position C, sorry, S, is influenced by the two left and right neighbors, and they are coupled. This is like a diffusion term. Okay. And from the simple model, when we use a kind of normal form or let's say similar function of the logistic map, we can have, depending on the value of A and depending on the coupling epsilon, here you have the numbers, we can obtain different patterns, like in the cellular automata, but here the variable is continuous. And these have different names, traveling waves. So here we will get traveling waves, they will move. Here we have like, I don't know if you can see, but here you have like clusters here. Then here you have like some speaky, noisy things. Here you have like turbulence. And you also can observe intermittency. Intermittency is when you have a periodic signal, and at some point you have a turbulent signal, and then it goes again to a periodic. It's like transient chaos. Okay, now I will skip this. I will explain very fast. So this is something that is very useful and has been very useful to study evolutionary process in silico. So actually, you have to imagine that you have a sequence of bits that can be, for example, a genome of a virus, or for example, can be a species that has some property. And when these bit strings can reproduce, can make a copy of itself, and every time it makes a copy, it is copying bit per bit replication, but we have some mutation. So at some point, when this sequence is copied, we can have mutations. Example here, okay? We can find mutations. And this is what we call the master sequence, the initial sequence that we can choose whatever we want. So we choose all zero sequence. And then at the end, we end up with a population of sequences with changes. And this is known as a quasi-species. And this quasi-species has been very important because we also have a theory behind this mathematical theory of quasi-species. Has been very important, as you can imagine, to study the evolution of viruses or processes related with evolution, okay? Immune system, pathogens interactions, whatever. So this is, of course, also an algorithm, simulation algorithm. Usually, well, it's like a Monte Carlo algorithm. You have digital genomes, these are vectors, and you have a population of vectors, and you can make replication, mutation, and you can evolve this system. This approach is extremely powerful, and I will show you an example, and this is what I was telling at the beginning, that we provided a qualitative explanation of a data with this model. And we observe the same in this simple system and in the patients of a hospital, okay? So advantages. This is also, you have to do a simulation program. You can use binary sequences, or actually you can use 4 letters alphabet like the DNA or the RNA. Of course, this is more demanding in terms of memory of the computer. But here, what we can do is to consider evolutionary processes and on top put population dynamics. We can put interactions between these digital genomes. So with this approach, we can track evolution. This is called in silico evolution. We can study evolution with a computer very fast. We can introduce stochasticity. We can introduce noise. we can introduce interactions at the genome of the level. This I will not explain because it's a little bit complicated. This is called epistasis. And epistasis is a very with a very simple example. This is something that happens in viruses, especially in RNA viruses. So RNA viruses like SARS-CoV-2, hepatitis C, Zika, they have the genomes are, well, SARS-CoV-2, it's like different because the genome is very long. It's 30,000 nucleotides, but other viruses have shorter genomes. So they have like 8, 9, 10,000 nucleotides. So epistasis is, I have a genome. Okay. Genome 1. I represent with bits, okay, but it doesn't matter. Okay, I introduce a mutation. And this genome has a very high fitness. It means that a virus with this genome replicates with probability 1. The fitness is very large. It's a virus that will invade the population of infected very, very fast. Then I take the same genome 2, and I introduce a mutation. And it turns out that now the fitness of this new virus is 1/2. Okay. Then I have another mutation. Here. Another place. And this genome has a fitness 1/2. So what happens if I have this genome? What is the fitness? If this one is 1/2 and this one is 1/2, this one, I don't know. But it turns out it should be 1/4, no? No, it's not 1/4. Is 2. Because these mutations interact each other. So I have one mutation here that decreases the fitness. I have another mutation here that decreases the fitness, but when I have the two together, the fitness increases. This is epistasis. These are not because these are genes. These can be genes that can regulate different processes. One can be activating, the other can be disactivating. But when they act together, when they are mutated together, the output is different. And there are a lot of examples of epistasis in viruses. I mean, the effect of mutation is not additive. This is what epistasis is. So I can study it very easily because I can consider, for example, that as I accumulate mutations, the more mutations I accumulate, and this is usually called fitness landscape. You have a fitness landscape that is, this is how we do, okay? There are other places, there are other ways on how we can do that, but what you measure is the fitness of the virus as a function of the number of mutations. This is the number of mutations I have. If I have straight line, the effect of adding mutations is linear, but there are other ways, for example, that this can be different. Adding mutations can have a more strong effect in decreasing the fitness or not. And these are different types of epistasis. And this has been studied a lot in real viruses. And you see that this exists in real viruses. Okay. Another thing I want to say is that when you have these kind of systems, you can make something that it's an open question in biology, that is the genotype-phenotype map. So one thing is the genotype, one thing is the sequence, and the other thing is the phenotype. Is how is this genotype affecting, for example, in the reproduction probability? And this is something that is not trivial in biology, okay? Because the sequence is not, so the sequence is what is under selection, and sorry, is what is under mutation, because this is what is mutating. But at the end, what is under selection is the fitness. That is how this organism is beyond the sequence. And one thing that we can do with this approach is that we can, a lot of times a priori or assuming, we can use, or let's say, I don't know how to say in English, we can give a given sequence, a given phenotype. Okay, for example, this one, probability of replication 1. Of course, I have mutations and all this stuff, but when I do these things, one of the things I have to assume is what is the fitness landscape, how mutations affect to the fitness of the populations. Okay, this is very fast. So this is, let's say, agent-based model. This is agent-based model. I can use, well, mixed populations. I mean, I have a population of strings that replicate, but I can also use this approach and put this approach in a cellular automata. How? I have some examples of this in my research. So you have a lattice, and now instead of having an individual, I have a genotype. And this genotype, when it replicates to a neighbor, you can have mutations, and you can introduce a lot of things. And I will show you 3 or 4 works that I did in my PhD where we studied coevolution with this simple model. And we saw quite interesting things. Okay, so this is very easy. These are hypercubes. Okay, so if we have an alphabet of 2, for example, 0, 0, and 1, if we have a string with L bits, then the total number of possibilities, let's say, is 2 to the power of L. Okay. What is a sequence space? This is a sequence space. This is for a system with 3 bits, okay, 3 bits, individuals with 3 bits. We can build a hypercube in which each node is one string and the neighbor is the same, but with a change, with a bit change. So this is a space in which we have all possible individuals. So 2 to the power of 3, which are 8. And if you see, these nodes are connected by single point mutations. And mutation will make the population to flow around these places. Okay, so if I have the sequence 0, 0, 0, and it mutates to 0, 1, 0, then the population moves here. This is a prey-predator system, which I have a predator. I have 8 different predators with 8 different genotypes that predate on preys with 3 bits. This is a very fundamental study, and what we did is that we have this system for 1 bit here, 2 bits here, and 3 bits here. And this is something that we used to study one of the most important evolutionary laws beyond Darwin, natural selection, that is, and this is a very exciting field of research, this is red queen dynamics. So the red queen dynamics, the name of the red queen is extracted from Alice in the Wonderland, the red queen. If you have read, there is at some point, Alice is with the red queen, and the red queen takes Alice from his arm, and they start running nearby a tree. They run very fast, but nothing is moving. Okay, and this is the image. So they are running very fast, but they are always here. They don't move. And this is a theory that says that actually the word that here there is a very funny history, because the guy that conceived this theory, the red queen dynamics theory, that, as I said, is very important, similar to the Darwin's theory of natural selection. he was not able to publish his research. No journal accepted this theory. What he did his own journal. He did a journal and published the first paper is this paper, and it's one of the most cited papers in evolutionary biology now. And this guy was a mathematician. If you want to read about it, it has a name like a heavy metal group. It's Lake Van Balen. It's not Van Halen, but it's Van Balen, okay? And this guy made a very important contributions in logics, fuzzy logics, mathematics. And well, I will not explain those history, but mainly what this theory says is that if you want to persist, if you want to survive, you need to move, you need to change. So you need to keep changing to keep in the same place. And I will give you a very simple example. You have a virus and the immune system. The virus is recognized by the immune system, and the immune system attacks the virus and kills the virus, okay? But the virus can evolve. So if the virus changes and the immune system is not able to recognize the virus, then the virus will persist. But if the immune system can also change, as it is, then it can change to trap again the virus, and you have an arms race. And this is why you need to continuous change to keep in the same place. Red Queen dynamics. So what we did here, very simple model. It's a toy model, but the results are amazing. This is a predator. This is a prey. And we assume that a predator 111 can only eat prey 111. The only prey that it can take is the same sequence that it has. And here we have mutation. It means that 111 can mutate to 110. So if the population of predators is all of the population 111 and the population of prey changes to 110, predators will not be able to hunt these ones. But predators also change. They also mutate. So these guys here are running all the time through the sequence space by mutation, trying to find the prey with the same sequence. So for this system that was proposed by my supervisor, Ricard Soler, when I was doing the PhD with him, he was very curious to see what happened here with the dynamics, with a very simple system. So we did different models. We did ordinary differential equation models with 1 bit and 3 bits, cellular automata with 1 bit, 2 bits, and 3 bits. And I have a PDEs model with 1 bit that has been I have been studying for 15 years, but I have not been able to publish it because I don't feel confident with the numerical resolution of the PDEs, but I will show you some results. So very interesting things. This is a paper that appeared in 2008. Here what we have is this very simple system with 1 bit, 0, 1. This is the predator, and this is the host, the prey, okay? So this species reproduce and the predators consume the same bit. And this is the model. This is very simple. This is a logistic growth. This is like a predator-prey model. This is the logistic growth of the sequences, I. Here, I is 2. This is the predation. This is this function. This is a Holling type 2 function that we explained the first day. And this is the predation. These are the preys, sorry, preys. Logistic growth, predation by the predators Y, but they can change between 0 and 1. And this is the mutation term or diffusion term that makes the species move in the sequence space and some decay. So this system has a periodic orbit. So you have this, you have to move to keep in the same place. So you have that prey escape, because they mutate, then predators change and they trap the prey, and then they start decreasing, but the prey change again and predators need to change, okay? And well, here we did some analysis of the hop bifurcation, etc. What happened when you put 3 bits? Here, this is a little bit more complicated because we have 16 ordinary differential equations with the same structure like this, but now I is 8 for X and A for I. And if I use the correct numerical method, I will ask Mark later. We observe chaos. So these are what we name the red queen attractors, red queen strange chaotic attractors. This is the dynamics for some parameter values of the population of prey and predators, summed all together, X, Y. And you can see here that you have this attractor that is very complex, and these are how the different genotypes fluctuate. Okay. We also have sensitivity to initial conditions, and well, what we did in this paper was study a little bit this thing. So what happens when we put space? When we put space, what I did, and this is another article. I don't know if I put here the reference. No. Well, it's a paper that was published in International Journal of Bifurcation and Chaos. And here what we did was something very easy that in some sense, I had to think how to modify, or let's say in other words, how to implement all these things going here in a cellular automata. Okay, so how do you do it? Okay, so here you have the growth of praise. So it's a logistic function. So I have a lattice. Okay. And here I will have, and here we did for the system, okay, like we use the three systems for the cellular automata. 1 bit, 2 bits, and 3 bits. So we did 3 different systems. So how I implement this term here? So I have preys and predators, and I have 1 bit. So this like this, with a matching, perfect matching. That is, predator 0 will only predate. So predator 0 will only predate prey 0, and 1 only 1. But these are not allowed. This is the length of the genome. So this is, if it is 1 bit, we have 2 individuals. If this is 2, we have 4 individuals. And if this is 3, we have 3 individuals. So the logistic term is just that if preys are X and predators are Y, so X with some probability reproduce. That's all. But since I have this diffusion term here, when they reproduce, they can mutate with some probability. So if I have the 2-bit prey and it reproduces with some per bit probability, it can change when reproduces. This is this term. And then I have some probability of dying. So this guy here, if I choose this cell, can die. And then I have the predator function. Okay. Here we didn't exactly implement the Holling type 2 function, because this is something that it's not trivial to do. But what I did is that if I have a predator, for example, 1 bit, predator 0, I choose a random neighbor, and if this predator 0 matches with the prey 0, this predator eats this one, so this disappears, and I put in a random place another one with some probability of mutation. And then that this probability of mutation, and then I have the possibility that this one disappears. So I only implement the rules. And to our surprise, we found that when you have 1 bit, these are the time series after iteration, similarly to the other way I was explaining, L per L times. We got like this periodic irregular time series with a spatial pattern. So all these things were moving all the time. And we did some smooth of the time series and some Fourier analysis. And here we had like kind of periodic orbit or torus, I don't know. And for some, when you have the whole populations, we have very complicated dynamics. And when we Yeah, for 2 bits, this is host parasites for the 2 bits for a given genotype, the 0000. And also we have very irregular. This is like a phase space. Okay, this is like a projection of the phase space with the hosts and the parasites with this sequence, and this is for 3 bits. And here you can see like this is not random. This is not a random thing. These are orbits that are turning, and they are quite similar to these attractors here. If you see this one, for example, you have these orbits that are going down and up. And here you can see something similar. Okay. Okay. And this is finally the PDE model. So yeah, I took the 1-bit system, the 1-bit ODE system, okay, if you remember, and I added the diffusion term. Here, what I did, it's like a finite difference method. So I implemented the ODEs with a 4th-order Runge-Kutta, and then I applied the diffusion term with a Laplacian operator with linear diffusion. Okay. I checked this way of doing so with a paper that was published with spiral waves and all this stuff, and it gave the same. So I assume it was the same, but this is not published because I am not so confident in the numerics. This is what we got with a cellular automata in which we have a space with 1 bit for the whole population, hosts and parasites. So if I take 1 bit and use this model, which is different because it's time continuous, this is a cellular automata, I got these things. These are some of the attractors. Okay, and these are the spatial patterns from 0y, 1y, 0x, and 1x, prey and predators. And I think this is spatiotemporal chaos, but I'm not confident about the numerical simulations. And well, this is a kind of phase space. It's not a phase space because PDEs, it's an infinite system. And usually what people do to show the dynamics is to use the embedding theorem of Takens, the delay coordinates. Okay, so this is all what I wanted to say about the different tools that we have for modeling. And now I will go to these 2 examples I was explaining to you. Qualitative explanation of a data with a very simple model, and quantitative explanation of data that I will explain in the 3rd session that I will do, because I will not have time. So for the qualitative modeling, this is, so when I was at UPF doing my PhD, some virologists there that have some expertise also in theoretical work, they have done the analysis of some patients that were infected with hepatitis C, okay, and they were observing something with the samples. They were observing different groups of infected people, and they come to talk to Ricard Soler, and he thought we could explain this phenomenon in the patients with one result of the quasi-species theory. Okay, now I will give you details. The other model, the quantitative, we will see in the next class. So a long time ago, there was a nosocomial epidemic outbreak of hepatitis C. So a nosocomial epidemic outbreak is when it usually happens in hospitals. Is usually you have an infected needle that is used for patients, and they are infected with a given virus. This is a nosocomial outbreak. So a needle with contaminated needle with hepatitis C contaminated 10 patients. Okay, the interest of these works, and the one I will show you, is that they realized very quickly about this contamination, and let's say they had the initial condition. So they know that some people were infected with this contaminated needle with hepatitis C, and they were infected from a single seropositive person. So in principle, the virus was the same for all these people. So they had a set of samples for 7 patients at an early time point, that is about 11 weeks after infection. And then they were able to follow the evolution of this virus in the patients. So what they observed? They observed so after this small amount of time, they observed 2 different types of patients. Ones with high viral load. This is the first sample taken by the patients. Here, patients are these numbers, okay, this code. So they have 4 patients with high viral load. This is the viral load. This is the amount of virus they have in blood. And this is the weeks post-infection. So they have 16 weeks, 37 weeks, okay? And then they also identified another set of patients with low viral load. So some patients had a lot of virus in the bodies, and another type of patients have a low amount of viruses. And what they realized is that for those patients with high viral load, they saw that the variability of the virus, this quasi-species, the different sequences, was lower than for the low viral group. So this is like a diagram with the nucleotides, different nucleotide sequences, okay? And in the patients with low viral group, low viral load, they saw that the variability of the sequences was much higher. So here there is a difference between these patients. I have to say that of all these patients, one of these patients, after some therapy, he or she removed the virus. Okay, the others were not able to remove the virus. And for the 3 patients with low viral load, since the viral load was very low, had spontaneous clearance. And this happens with hepatitis C virus. I think that 15% of the patients remove the virus by themselves. So the question was, can we do a simple model to reproduce this effect? High viral load, low quasi-species variance, low viral load, high quasi-species variance? This is a very hard question. Other things, what they saw, so they quantified. For the high viral load and the low viral load, they computed entropy measures, Shannon entropy. And they saw that for the high viral load, the Shannon entropy was low compared to those of low viral load. Okay, this is between 0 and 1. So more or less the Shannon entropy for the people with a large amount of viruses was about 0.03, 0.4, 0.5, and for the low viral load, 0.8, 0.80 something. Okay, since Ricard had been working with quasi-species theory for a long time, and I also started doing my PhD on quasi-species theory, we thought that we could interpret this result in terms of the quasi-species theory. Okay, so the idea is that, so the result or conjecture that we did is that this origin between the inverse correlation between the viral load and the quasi-species complexity could be explained with a very simple model. And it was a problem related on where the quasi-species was located in the sequence space. Okay, so sequence space. If I have a sequence with 2 bits, okay, so the sequence space with 1 bit, sorry, the sequence space is 0, 1, is 0, 1. Okay, if I have 2 bits, then the sequence space is like this. 0, 0, 0, 1, 1, 0, 1, 1, okay? 1 neighbor mutations. A virus, SARS-CoV-2, SARS coronavirus 2, has 4 to the power of 30,000 possible configurations. This is something crazy. And this is the sequence space also. It's a multidimensional space. where each genome has a neighbor with single mutation. Very simple model. If you remember, in my first lecture, I was talking about a model in which we have a master sequence that was reproducing under some mutation. This is the outflow term, if you remember. This is, let's say, the master sequence that in our case is the first infecting virus of the patient. And then we group. This is a very, very simple, the simplest model that you can do, a toy model, but with this model, we explain this thing. And then we have all the pool of mutants, all the possible mutants in another compartment that provide or that come from the mutation. Here there is the replication rate. This is the error-free replication. So here I need the error replication, mu replication mutation rate, plus some replication of the mutants minus the outflow. So what is this model considering? This model is considering a single peak landscape. Single peak landscape is that we have fitness, number of mutations, 0 mutations, we have fitness Fm. Any mutant has a low fitness, Fm, fitness of mutants, that's all. These are the equations. This is a very simple model. It has been studied a lot. Here, X1 is a master sequence, X2 is the pool of mutants. And our conjecture was, we have two possibilities. One thing I have to say is that one may expect that the mutation rate of the hepatitis C virus should be the same in all the patients. Okay? The mutation rate more or less is the same. And we had data on mutation frequencies, and they were very similar. The difference is that we consider could be happening between the two patient classes is the ability of the virus to replicate. Okay? Or because the immune system was different and the virus was not replicating so much. So our conjecture is in the patients with low viral load, okay, that we have more quasi-species complexity, the master sequence has mutated and replicated a lot and is occupying a lot of nodes of the sequence space. Low viral load because the population of master sequence is low, but the number of mutants increases. For the high viral load, the master sequence that it's the initially infected genome or virus is very large, but it is more located in the sequence space. Okay? It has like remained close to this node. One important thing, here in this model, one can see the so-called error threshold. This is a very important concept in evolutionary virology. And actually, what I am now going to draw in the picture, in the blackboard, has been found with HIV in experiments. There is a theory that is very related with the quasi-species theory. It's one of the main results of quasi-species theory, is that, well, no, this would be the quasi-species. The quasi-species population. In our case, master and mutants. So there is a result that says that as mutation increases, the number of the master sequence decreases, the number of mutants increases, and at some point, well, it depends, and at some point, the master sequence disappears, and the population is dominated by mutants. And this is the transcritical scenario, So if you do simulations with bit strings, with this landscape, what you see is that the per bit mutation, and here we have master and mutants, but now I do a bit string system. And I have the master sequence, for example, this one, and then all these mutants are any genome with any mutation, with one mutation, two mutations, whatever. So what you see is that the 0, 0, 0, 0, 0 sequence goes to 0, and here you have like the spectrum of mutants. Okay, so these are the mutants, this is the master sequence. This can be achieved by tuning mu, but it can also be achieved tuning the fitness. Okay. Why? Because we have two equilibrium points. We have the origin and this one when we have coexistence. And this equilibrium point depends on the fitness of the master and on the mutants. So I can achieve the error threshold by tuning the replication. And in our patient samples, we assume that mutation was the same. So our conjecture was in these patients, the virus is replicating differently. And this is why we see these patterns. Okay. So this is the model. This is the typical error threshold where the master sequence decreases linearly. It's actually this line here with mutation, but the error threshold where the master sequence disappears and the mutants dominate the population can also be achieved by decreasing the replication rate. So here we have the quasi-species because we have, let's say, the master sequence, but this bifurcation curve, the master genome disappears and we have the mutants. Okay. This is the simulation with the bit strings. The same system. We have sequence with a big fitness and all the mutants with the same fitness. This is an oversimplification of viruses because viruses do not have these landscapes usually. Okay, there are differences with when you study the role of mutations on the fitness of viruses. But we use the simple case. And this is what we have, what you see here. In the thick line is the master sequence, and the thin lines are all the mutants that appear with a mutation rate of 0.04, fixing the mutation rate of the mutants and decreasing the replication rate of the master sequence. This is the error threshold. So here there are no more master genomes. Okay. Can we explain those results with this model? Yes, qualitatively. Why? Because what we did is to compute the stationary entropy of the whole population by decreasing the population size, by decreasing the replication rate of the mutants. So what does it mean? It means that if we, of the master sequence. So what does it mean? Look, here we are decreasing the replication rate of the master genome, and the population is decreasing. If you remember what the people in this hospital, that was the Figueras Hospital, observed, is that those patients with low viral load, viral load of the initial virus, had higher variability. So what happens if we decrease the replication rate of the master genome? That we go to scenarios of low viral load. But does the variance increase as F1 decreases? And the answer is yes. As F1 decreases, the population, it's more difficult for the population to remain here. It's more difficult to remain here, and it spreads on the sequence space. And this is what we found. We found an inverse correlation between viral load and quasi-species complexity. That is, the low the viral load, that would decrease. If we put here an axis with a viral load, it would decrease, and the stationary entropy increases. This is mainly the Shannon entropy. Okay, so we were able to reproduce these clinical results with this simple model. Okay, here are a couple of comments. Is this happening in the patients? Exactly this? I would say no. Okay, I would say no. Is it possible that this relation between the replication rate of the virus and the increase in the entropy was happening in the patients and is also happening in our model? I would say yes. Is this model reproducing the clinical data? I would say yes. But is this model realistic? I would say no. Okay. But I mean, probably, and this is something I want to say, and this is something that many times we see, is that imagine that we complicate the system a little bit. I mean, in terms of the landscape, for example, we assume other classes, of the other mutant classes, and we make the model more, let's say, more realistic in some way. I am pretty sure that this effect would be also observable. So this is the simplest model we can do. It is very unrealistic for a population of viruses, but because of the characteristics of this system, of the virus, of how the patients were infected, and the evolution of the time, the control of the samples, etc., this simple model reproduced quite well the expected results from a clinical point of view. This is a paper that we published in Journal of Theoretical Biology, and I think it's one of the only articles in which this fitness landscape has been useful for something in the real world. I mean, because this fitness landscape is a very simplified landscape. So viruses do not behave like this. But for this system, at least, it worked. Okay, so next session that I will give, the 22 of November, I will explain the quantitative work that we did to measure the production of genomes in a virus using a simulated annealing. Okay, and from this part, then Luis will go on with the techniques for the optimization. Okay, any question? Any comment? No? From those online, is there any question or comment? You can turn on your microphones. If anybody has some question, you can use the chat or the microphone. Okay, so no questions. If any of you have any question, you can send me an email or we can talk in the other session. So thank you very much. I hope this will be interesting for you, and see you in the next session. Thank you.